this video, we will describe the results screen for exploring the data extracted during image analysis. There are two ways to access the results screen. Firstly, it appears following image analysis of a dataset, which is what we can see now. A separate video will demonstrate exploring the raw images by accessing the results screen with a second method, which is directly clicking on the Results tab at the upper right of the screen. As we look at the results screen, first and foremost we see the heat map in the upper center portion of the screen. This map has the same layout as the plate used for image acquisition, and the colors depict a relative visual scale for the values in each well of the selected property value, which is chosen through the menus on the left. The color range spans the minimum and maximum values of the quantified property and are shown in blue and red respectively. Colors corresponding to mid-range values are set automatically according to the distribution of the values across the plate. The heat map is intended to provide users with a quick readout of the quantified data across the plate to visualize the effect of experimental conditions. The data visualized through the heat map is chosen using the menus on the left. First, we choose the statistic that we would like to use for analyzing our data. Operations include the average value per well, the percent of a population falling within an upper and lower bound set using the histogram, the count of the cells, or a ratio of the extracted properties. Currently, we're calculating the average value over all cells within each well. Next, we choose the property, also known as attribute, that we want to analyze and visualize. It's important to note that the properties quantified and selected through this menu depend on the application used for image analysis. The current menu corresponds to the protein expression application, which was covered in the previous video describing the calibration of analysis parameters. We see that the total integrated fluorescence intensity in the protein channel is selected, and in this analysis corresponds to the red channel. To change the attribute visualized, we simply click on it in the Property menu. We see the heat map update to display the average mean intensity for each well, color-coded according to the color bar on the right. If we click on a well, the software returns the value for that well in cyan-colored text below the color bar. As well, because we have defined positive and negative controls in our experiment, we have experiment statistics such as the coefficient of variation, CV, the sector boundary, SB, and the Z prime values that correspond to our data, and they are displayed here to the right of the heat map. Below and to the left, we see a histogram showing the distribution of the currently selected property for all cells identified. To the right, we see a few check boxes that enable selection of a portion of the plate to include in the histogram. Currently, we are looking at data from all wells within the plate. By selecting a few wells on the heat map, we can visualize the histogram of this subset by first deselecting the full plate histogram, and secondly, clicking the box for the selected region, which updates the histogram. We can also specifically visualize the positive or negative control wells. Below the histogram, we can see fields where we are able to set a lower threshold and upper threshold for our gating statistic. The upper and lower bounds can be set using the interactive buttons or by clicking on the field itself and typing in the number manually. As we do so, we can see the vertical pink lines change position, indicating the position of the lower and upper thresholds respectively. Having set the gating thresholds, we can select the gated population statistic, then select the mean intensity attribute for which we set the thresholds. The heat map updates to display the percentage of cells in each well that fall between the thresholds. To the right, we see a two-dimensional scatter plot where we can select two properties to plot against one another. We can change the properties included in the plot through the Scatterplot Edit button. Within this menu, we can select the two properties of interest. As well, we can select subpopulations if we've defined them, and thereby visualize the relationship between the selected properties per object. Similar to the histogram, the scatter plot can be generated for the full plate, for a selected region, or for the positive or negative control wells. Because this dataset was acquired and analyzed with an uploaded tags file, the dose curve tab is activated and accessible. The tags file, which is described in a separate video, includes information regarding treatment conditions within each well of a multi-well plate. Through the dose curve, we're able to plot the currently selected property versus the concentration of the treatment applied in order to get a quick visualization for the dose response in our experiment. To generate the curve of interest, we first select the property that we would like to have plotted versus concentration. Then, we use the drop-down menus below the plot to select the treatment we would like to visualize along the horizontal or x-axis. In this case, it's the nicotazole treatment. We can then choose to have a linear or logarithmic scale along the x or y axis using the log button. Optionally, the data can be fit with a hill curve using this checkbox above the plot. Datasets acquired as time-lapse give access to the time curve tab, which here is darkened out. To access it, we will briefly look at results from a dataset that was acquired in time-lapse with Z-Stack imaging. We see that above the histogram there are additional menus for selecting specific time points or Z-slices of interest. Currently, we are viewing data from all time points and all Z-slices. If we click on different time points and/or Z-slices, we can see both the hitmap and histogram plots update to reflect the data present only within the indicated time points or Z-slices. There are only 2 wells in the hitmap, but the updating can be seen by the refreshing of the color bar and its min-max values. On the bottom right, we can now see the Time tab activated, meaning that we can access the time evolution plot. Within the time-lapse plot, we can see how a quantified attribute changes over the course of a time-lapse acquisition. Similar to other plots, we can change the property being plotted and select a subset of wells within the plate, shown in cyan, with respect to the plate-wide trend, shown in dark green. Now let's return to the first dataset we were exploring. The data visualized in all of these plots can readily be accessed and extracted simply by clicking on any of them and selecting either the Copy Values or Export Values option. A more powerful and comprehensive method of saving such data is to simply click on the Report button. This button generates the report that you see here that can optionally be saved as a PDF to facilitate keeping of a digital lab notebook. As we scroll down, we can see the summary of both the image acquisition and image analysis parameters that were set for this dataset. Farther down, we see the data visualizations that are currently displayed on the Athena results screen. The hyperlinks shown in blue will give access to comma-separated value files containing the values plotted or visually represented within each plot. For instance, selecting the hyperlink below the hitmap graphic opens the statistic.csv file showing the average cellular intensity value quantified per well. Clicking the See All Objects hyperlink opens the master data.csv file, which contains all of the metrics quantified per cell. The information and results contained within the report file are accessible for offline analysis and can be accessed through the analysis folder on the hard drive or network drive. The easiest way to access it is through Athena using the hyperlink at the upper left of the software shown here. Clicking on the link opens an Explorer window with the path set to the current analysis folder. The data that has been generated is contained within the reports folder. Opening this folder, we are able to see a sequentially numbered report folder that's created every time we click the Report button. The Report 0 folder is created by default, with the sequentially numbered report folders generated every time that we click a report button in Athena. Here, we can also see the data.csv file that is the master table of all data quantified from individual cells detected within each image acquired on our multi-well plate. Opening the Report 1 folder, we see an HTML file which can be opened in any internet browser program to recreate the report that we first saw in Athena. All of the quantified data visualized in the plots is present in the CSV files located within the Reports folder. These comma-separated value files can readily and easily be imported into other software for further analysis or plotting. Lastly, all of the data visualizations shown in the report HTML file are contained within this folder named Hermes Report folder. Opening it, we see the JPEG images containing all of the data visualization plots we saw in Athena. These can then be used directly in presentations or digital lab notebooks. Thus far, I have described the data exploration and visualization available in Athena following image analysis. Accessing the results screen from elsewhere in Athena, such as from the loader screen, will be described in a separate video.